Thank you, BIOMIN, for having me back. I was thinking about on my way over from the airport how many years I've been coming to this meeting. I think I've missed 1 year and about 20 now, so it's kind of a little bit of a homecoming. It's good to be back in Indiana. So thank you to the BIOMIN folks for having me here. Today I'm going to preface this afternoon by talking a little bit about the gut. And I have to thank Mike. Mike's in the room. Hey, Mike. For fostering my interest in the gut. We've had a lot of good discussions over the years of its role and where it has missteps, at least. Now, the topic I'm going to talk about is mainly with mycotoxins. And I didn't necessarily mean to get into any mycotoxin research over the years, but I looked at it more as one of those stressors and one of those unknowns, at least, of what we do not know, at least as far as translation of effects on functions within the gut itself. So that's kind of the format, at least. And before I get into mycotoxins specifically, I wanted to just review the gut and what we know, what we don't know, what we're doing on the research side collectively as a community, maybe some things we are not doing so well as well. And we've made this huge transition this past year, at least on the broiler market, in transitioning to AGP-free production. And as so, we're having a little bit more of necrotic enteritis come about. And so I wanted to talk about really the pathogenesis of that process of what we experience with necrotic enteritis. First and foremost, we have some colonization that's occurring predominantly from cocci. We have replication and multiplication within the digestive tract. We have acquisition then of different nutrients for both cocci as well as perfringens. They then have different mechanisms to evade the immune response, causing extensive tissue damage and then further transmission through fecal-oral routes. The one thing that we don't necessarily do so well is look specifically at interventions that might target very specific roles within this process. And the other one that's not on this slide as well is we don't necessarily think about compounds that may help at least in the tissue repair and recovery post this cycle of pathogenesis. And there may be roles specifically for intervention strategies for those compounds within this cycle, but then also afterwards within the recovery process. So a number of you have seen, so this is not necessarily new, but as we think about this acquisition of nutrient phase is very critical to the pathogenesis that does occur. Oftentimes we think of this as excess undigestible protein, but it's important to realize that we often ignore that change in translocation. We heard Mark talk about that just a little bit ago, but that change in translocation goes both ways. Not only from those pathogens gaining entrance directly through those pericellular barriers into the gut, but then also leakage of those plasma proteins into the lumen of the gut. And it's that leakage of that very readily available nutrient source, whether it's from plasma, blood, that then kicks off this cascade of biofilm development for that perfringens within the lumen. And it's important that we recognize that process as we think about this cascade of how it's affecting the bird and how that disease then progresses. So we know the predisposing factors, at least for necrotic enteritis, we have the cocci infection, we have removal of those AGPs, diets rich in non-starch polysaccharides tend to increase through changes to mucin production and mucin viscosity, and at least undigested protein potentially coming in to the gut itself. We also, as I just mentioned, that change in plasma protein leakage, but what we really don't know is really what role mycotoxins play in this check and balance system for recognizing that those pathogens are there and the immune process to go forward with at least clearance of those pathogens. So oftentimes, you know, we picture this as the iceberg. When we're thinking about mycotoxins, we're typically thinking of them as those clinical lesions that we can actually see. It's that unrecognized portion, though, that subclinical portion that often goes unrecognized that will be potentially impacting performance or other changes in the bird that we are unaware of. And it's very difficult to go forward and recognize those when we don't visually see the lesions in the field. And the science behind where we are on mycotoxins come leaps and bounds over the past decade, where we're just gaining a foothold in recognizing what biomarkers, whether it be from physiological changes, toxicological changes, changes to immunological functions that are very distinctive to different mycotoxins. So let's get back to the gut. Let's talk about it. And for a number of you that have seen me give these talks over the years on the gut, this is a very familiar slide, at least. We know that the gut is a very nutrient and energy rich tissue and that consumes approximately 20% of dietary energy. But more importantly, I tend to think of it in terms of protein. In terms of protein, its turnover rate is estimated to be 50 to 75% per day, and we'll talk a little bit more about protein turnover here in a little bit. But essentially then 1 out of every 4 proteins produced is actually synthesized and secreted into the gut over time. So as we think of roles and where potential mycotoxins may interface with the protein production standpoint, there's a whole host of places that we know that mycotoxins can interfere with that protein synthetic process, whether it be from the aflatoxins and the trichosynthenes interrupting DNA template or RNA transcription with aflatoxin, or translation itself, whether it be affected by aflatoxin, the trichosynthenes, or ochratoxin, or through specific metabolic reactions through a range of different mycotoxins. We know that these particular mycotoxins tend to interrupt these different places in overall protein synthesis. And as such, that process becomes much less efficient in our highly producing, highly reactive animals. So if we think of the gut as a barrier, we need to think of it as to what roles and different functions it has. From a physical barrier, we know that that first layer, that mucin layer, traps and neutralizes bacteria. It's constantly turning over. Some microbes, such as perfringens, are mucolytic, which tend to eat that mucin and actually stimulate mucin production as well. But it is that, first and foremost, that protective layer. Secondarily, we also have that turnover of those cells that changes and has different changes to turnover rates. We often think of that as being a very static process, but it has a range, and we'll talk a little bit more about that process here in a minute. But then also those barriers between the cells, those tight junctions, and we'll talk specifically about some of the tight junctions and roles and functions that some of those mycotoxins have to disrupt those tight junctions within the gut. So again, this process of renewal, and we'll talk about the renewal process, involves the production of new cells, which we think are more isolated down to the crypt region, and as they mature, they migrate up the crypt-villus axis. They tend to differentiate in their functionality. Think of that from the different transporters, different membrane-bound enzymes that are produced, before they then are sloughed from the villus tip. And this turnover process is essential for that integrity of the intestinal epithelium. Now this process becomes disrupted at some points in time, particularly when you're faced with a very overt challenge. And I like always showing this slide back to 1973 to just really illustrate how dynamic this process is with an overt infection. So in this particular study, they infected birds with Emeria cervulina, typically affecting the duodenum. Here you see with the light blue bars, the control birds having a turnover per day of 10% of that tissue. With the overt challenge of Emeria cervulina, they had 30% turnover of that tissue. A 300% change in turnover rate of the dynamics of that system. And as you would imagine, the maintenance needs for that particular tissue go through the roof. Now, while we know that Emeria cervulina is mainly affecting the duodenum, it had trickle-down effects on the turnover in the jejunum as well as the ileum. Jejunum, the control birds having about 15% turnover, and controls, again, about 10-12% in the ileum. But you see the trickle-down effect on that turnover rate being about 80% higher in the infected birds in the jejunum, and in the ileum about 90% more. The body's trying to respond with those infected cells by essentially trying to rid itself of those infected cells more readily. Some of the work that I had done probably a decade ago with the folks at BIOMIN, we were looking at some of those subclinical challenges and changes that occur within those subclinical challenges. And we were developing a model at that particular point in time where we had our experimental pens. Day one, we would feed a heavy dose of paracox, in this case, coccidial vaccine, to half of the birds in the pen. And then we'd sample birds and the other half of the pen later on after we knew that that cocci had gone through its cycle within the litter itself. So what you see here is these birds at 27 days of age, those birds that were not given the vaccine on day one. And you'll see that essentially those birds given the 5x dose of paracox versus the 1x dose, we had a 20% difference in turnover rates just because of that increased pressure from oocysts present within the litter, which was always amazing to me. You did not see very overt lesions at all, but yet that 20% difference is quite a difference in terms of maintenance function to that tissue. We did some other studies later on looking at a 10x paracox dose here at 5 and 7 days post-challenge, just to give you an idea of what it does to that overall tissue dynamic. You can see very high turnover rate in these, in what we would expect at least from a severe stunting, at least in a heavy coccidial challenge, versus that recovery phase 3 weeks later, 21 days post-challenge, where you can see that tissue has then recovered. And this dynamic, at least as we talk about different feed additives and their functions, again, we need to probably target the times in that bird's life when they are needing to go through that, dealing with the challenge, but then also recovering from and repairing that tissue. And we need to recognize at least the time that it takes to fully repair that tissue once that challenge has been left. So I'm going to touch now a little bit more specifically on mycotoxins and mycotoxins' role in that of the gut. And as you had seen before, I think of this in terms of an iceberg, really, and as we think of mycotoxins, oftentimes we think of this as those overt lesions, which we'd expect from mouth lesions with the scarpinols and the T2, the fatty livers from aflatoxin, kidney damage from ochratoxin, or in the case of the feed mill, when you're seeing those different fungi and molds being present. So Bertrand Grenier spent about three plus years in my lab at Purdue. Bertrand came from one of the labs in France, one of the governmental labs, where he had done immunological work in the pig with mycotoxins, and he wanted to cross-train and essentially learn the same thing in the case of the bird. And as one of those preparatory exercises, Bertrand put together essentially a question, spawned this: can mycotoxins affect the gut? And we were wanting at that point to really dig into the literature to know at what point we were in roles that different mycotoxins play in their role on the gut. So he did this meta-analysis across all different livestock and poultry species. And the interesting thing, he split this apart by studies as to their effects on the animal, from nutrient digestibility functions, nutritional functions, enzyme activities, nutrient uptake, changes to flora, but then also defense functions such as barrier integrity, mucosal immunity, and actual pathogen challenges with clearance. The biggest number of studies, 32 of these studies, were done with Dioxin of Allinol. And a lot of those studies were done in the case of the pig. So a lot of what we know, at least in terms of dioxin of allinol's role and effects on the gut, have been translated from the pig. And there's some differences from the pig to that of the bird, and functionality differences that we need to tease out. And since this point in time, this was published in 2013, we had continued on some of that work at Purdue, and there's been some European work as well that's dug into those roles and functions with those field-based mycotoxins, specifically dioxin of allinol as well as fumonisin. So let's talk a little bit more about those. But to put it in perspective, I stole some slides from Raj. Thank you, Raj. Just to give you perspective on what we're dealing with in terms of dosage. The other thing that becomes very simplistic in our minds as well is you look at the literature, it's very easy for us to categorize things by compound X has function of Y. And we often ignore concentrations and how those concentrations translate to an effect. And those concentrations are very important to think about in terms of their role and what they may translate into that effect on the animal. So this is partly why I put this on this slide, just so you can have a perspective of where we've been most readily. Here in the upper Midwest, over the past few years, as we've changed our cropping practices, going from rotation with corn and soy to all corn, we've also gone to no-till, which has allowed essentially a buildup over time of some of those field-based mycotoxins that have slowly kept a baseline going forward. So if we look at occurrence and distribution of feed ingredients throughout the U.S., 75% of those samples that they collected were positive for DON. The average of those positives was 1,670 parts per billion and a max of 11.7. So let's think about that. We know in the case of the pig's very sensitive. That's one of the reasons that dioxin of allinol is also named vomitoxin, because of its vomiting effect that it has on the pig at levels roughly about 1 part per million or less. So we're very much within that range here in the means of the positive. So it's no wonder that in the Midwest we're keeping an eyeball on where that is. Within Bertrand's paper, he broke this down within the literature into what we would call realistic dosage, those that are very commonly occurring doses out in the field, doses on very high peak years, he would call occasional doses, versus that of unrealistic dosage, which researchers at universities, myself probably included, at some point fed the heck out of it just to see how it would respond, right? So it's important to keep those doses in mind as we have much more data now to go back to have a better understanding where those realistic doses are on a year-to-year basis. So if we're looking at dioxin of allinol, we're typically talking less than 5 parts per million, occasional doses of 5 to 25. Here I put, again, that last cropping year's levels for DON. For fumonisin, I'm also looking at these over here. Those realistic doses being roughly a little bit less than 10, and occasional doses anywhere from 10 to 40. Okay? So within the US, just to have a geographical representation as to where we're seeing these levels, and the darker red was samples over 900 parts per billion. So here we are within the upper Midwest, all of those samples typically being above the 900 parts per billion. Again, I'll talk in here a little bit of what that really means as far as translational effects on the bird's response. The pink states are less than 900 parts per billion. Fortunately, Georgia's there, but unfortunately, we get most of our corn from Illinois, Indiana, and Ohio. So if we think just about absorption of those mycotoxins as to where they're occurring, it's important to realize, again, most of our base, at least for understanding mycotoxins' role, on the effect of the gastrointestinal tract is being pulled from that of the pig. We're filling in those gaps, at least on where the differences are in the case of the bird. For that storage mycotoxin, aflatoxin, roughly we're talking very high levels of the absorption, over 80%. But as we look at dioxin of allinol and fumonisin, in the case of the pig, for Don, we're seeing absorption rates of about 55%. In the case of fumonisin, about 3 to 6%. But in the bird, we often had to ignore them, largely because the absorption rates are very low. Okay, in the case of Don, we're only talking 5 to 20%. In the case of fumonisin, we're talking less than 1%. So it'd be easy to essentially ignore them. Say, they must not be having an effect, right? But the question comes to, what is the role of that unabsorbed part, and is it still having a role and function on how the intestinal tract is responding? For fumonisin, one of the neat things about moving to Athens, Georgia, is that the USDA ARS unit there close to campus, they've had a group that have spent their entire careers, a few of those folks have retired, where they've essentially looked at physiological roles of fumonisin over the years, and they were one of the first to essentially attribute biomarker responses to that of mycotoxins, and they did that specifically with fumonisin. And the biomarker that they teased out within this group was really this interruption of sphingolipid metabolism. And the question comes to here in a minute as to, well, what does it matter? Sphingolipid metabolism, right? So how does that translate into other effects? So we know that fumonisin, just because of its shape and size, it fits very similarly to that of sphingonine, and so it interrupts and plays havoc with this CoA-dependent ceramide synthase, and therefore you can measure the changes in ratios of sphingonine to sphingosine, and they're very indicative of changes that occur upon exposure to fumonisin. So one of Bertrand's first studies that he did with us at Purdue was really to characterize how different concentrations at different times of exposure would translate into how these biomarkers were translating into effects. If we look at serum, not much of a response in terms of the ratios, but you can see upwards of 50 to 100 parts per million, we're starting to tick up those changes to fumonisin. If we looked at the same literature in the case of the pig, we would see that much lower levels tended to kick that up, partly because of that increased absorptive capacity that is in the case of the pig. Similar as you would think about that, showing somewhat of an indication that you're having some disruption amongst that barrier of the gut or absorption within that gut. Kidney, very high concentrations, having that translated effect, as well as the liver. But what really stuck out to us was that of the gut. In this particular study, we were seeing concentrations as low as 20 parts per million in the jejunum. Similarly, in the ileum, we're having these much larger effects on this particular biomarker of exposure. At the same time, because Bertrand was a very industrious young postdoc, he wanted to try and analyze all sorts of things. He had interest in immunology, and so at that point in time, he also started pulling samples and looking at inflammatory response markers, and across the numerous different inflammatory markers, he was seeing an uptick in inflammatory marker status in those ileums from birds fed those higher levels of fumonisin, even as low as 20 parts per million. And that was kind of a huh moment. You often have one of those huh moments. And that meant, as a researcher, I'm always looking at that, well, that's interesting. But does that really matter? So yes, they have increased inflammatory markers, but does it translate into any other effects or susceptibility of those particular birds? And that's where we dug into the literature a little bit more and tried to design some other experiments to really answer that does it really matter question. Okay? So we know, based off of some other work that others have done over the years, this is from mainly from some folks at the University of Vienna, some folks in Brazil, some folks in Michigan, some folks in Belgium, looking at Dioxin of Allinol in cell culture models. We know that permeability to the gut is drastically changed through changes in tight junctions, which we don't, at this point, didn't really know why, but also changes to nutrient transport, specifically glucose, by interruptions on transporters both on the apical as well as the basolateral surface. And that had translational effects on how flow of nutrient was actually going from a active uptake standpoint. Along came another student, and one of those questions came about as to, well, how is that interruption to that barrier actually occurring? And she worked with Kola Adjuan and myself here the past few years. She's going to defend her PhD here in a week and a half, 2 weeks. Yeah. So it's very exciting. She did some really neat research trying to tease out the mechanisms of what we saw and thought about those things that were occurring. So in these cell culture systems here, these were IPEC-J2 jejunal cells from pigs. And she essentially created a similar baseline to what was in the literature, just to replicate and understand. So here we see increasing concentration over time, 1 hour, 24 hours, 48 hours of exposure. Changes to transepithelial resistance, and as you can see, as early as 24 hours, we see dramatic reductions in resistance of that tissue. 48 hours, we see a little bit lower dose having a little bit more of an effect. And to understand then how that affects translocation, we began looking at a fluorescently labeled dextran and passage across that particular tissue, or cell culture in this case. We can see dramatic increases to passage of that dextran across the gut. Not anything different necessarily than what was in the literature already. But what we came to find out, and as she's dug into changes, at least from mRNA for particular families of tight junctions, here we have the schematic over on the right for different tight junction families from the zona occludens, the occludens family, the claudin families, and the E-cadherin families. We found that the main one that was being affected in her cell culture model, at least, was that of the claudin family. And it was very interesting. So for the grad students in the room, when you're only measuring mRNA and message with real-time PCR, it's hard to get a full picture of what's going on physiologically without understanding protein synthesis and also protein degradation. And this is one of those stories that's important to think about, that full picture as well. So if she was only measuring message, here you see with the control birds, not much change in those claudin families. This one's specifically claudin 1 over time. But as you can see, if you're only measuring message, you can see the Dioxin of Allinol had a huge uptick in message of this particular tight junction within a very short period of time, within as little as 3 to 4 hours. Now when she started looking at protein, and unfortunately in the case of the bird, we don't have all the analytical tools quite yet to do all the same types of science as what we can do in the pig. But in this case, when we were looking at this particular model, we began looking at what was happening with the protein. And essentially what we were seeing is in as little as 3 hours here with the control birds versus those that were challenged with Dioxin of Allinol, we were seeing a dramatic reduction in as little as 3 hours of that particular claudin tight junction, and it was very similar across the entire family. So protein degradation, it seems that it was targeted somewhat specifically to interrupt that, and what we think of as the protein degradation pathway that's occurring is very much upregulated by Dioxin of Allinol, such that message itself and production of message tries to respond, but it can't compensate in a timely fashion to overcome that protein degradation that's occurring. So let's get back to the what does it really matter question, okay? So as we look at this, we formulated some other studies where we began looking at a coccidial challenge model. And in this case, we were just using a heavy dose of a coccidial vaccine. So to familiarize yourself with where we were here with Dioxin of Allinol, fumonisin, and the combination of the two, back to somewhat occasional dose, somewhat realistic dose, just depending on where you are and what the year is in the US. 2 parts per million of DON. Fumonisin, we were at 20 parts per million, and the combination of the two. So looking then in the challenge birds, just scraping mucosa, looking at numbers of oocysts. You can see the control birds here, a little bit of cocci, but nearly double the amount of oocysts present in that mucosa in the birds that were exposed to dioxin of allinol and fumonisin. For counting oocysts in the excreta, fumonisin alone or the combination of the two, really there was an uptick in oocyst numbers present in those treatments. We also began looking at the biomarkers to see what was happening, and between the white bars here are the unchallenged birds, the dark bars are the challenged birds. The interesting thing was we were seeing an increased effect here of the combination of the two with that biomarker being present within the serum, meaning that it looks like it's passing through, then having more effect in those challenged birds in the liver, partly again because of passage through, and then translational effects on the liver. However, in the ileum itself, we saw more of an effect in the control birds, but not as much in the fumonisin alone or the combination of the two, partly because we think more of that fumonisin is probably getting through, translocating through the barrier of the gut, then having translational effects on organ systems rather than directly that unabsorbed part on the lower ileum itself. So this was a little bit of, an answer to that question that Bertrand had early on was, does it really matter? Well, it looks like it is. It looks like there's more oocysts within the tissue itself, more oocysts coming out in the excreta, potentially more damage to the tissue itself. He also came over to Worcester, did a little bit of work with Ramesh, trying to understand mechanisms then of what was happening, at least from an immunological response. And Ramesh could answer this and talk about it at length, much more than I could. But my take-home message from this, and essentially the challenge birds, control of that inflammatory process through the Tregs, the Treg lymphocytes, took more to essentially control that challenge in those birds exposed to dioxin of allinol and cocci. And we know from literature, and I'll show you a little bit of the literature here in a minute, severity of those lesions, extended duration of that pathogenesis to that disease was extended in the case of exposure to DON, fumonisin, or the combination of the two. The other thing that was interesting, and Gunther Entoson, who was PhD student at the University of Ghent and has graduated and continued on in a research scientist capacity, was doing very tandem work, so to speak, at University of Ghent in Belgium. And he's done a number of very eloquent studies. As we were doing our model, he was doing somewhat of a different model, looking at necrotic enteritis, but then also other physiological roles and changes that were happening within the bird itself. So very similar type of design to what I just showed you with our cocci study. Here, these are just non-cocci challenged birds in this particular case, 4.2 to 4.6 parts per million of DON, 15 to 19 parts per million of fumonisin. The stress responses of these birds were very, very different as well. We see that corticosterone levels in the mycotoxin exposed birds were much higher than those versus that of the control. Now, they have a necrotic enteritis model A little bit different than some of the others we're probably used to at Southern Poultry Research or at CQR out in Colorado. For this particular one, they used a field-based perfringens strain, and very similar design here. Negative control, Dioxin of Allinol alone, unchallenged birds for the first 2 bars. Second 2 bars, the CP is challenged with Clostridium perfringens. The CP plus DON, that's 3.8 to 4.4 parts per million of Dioxin of Allinol. You can see that with the Dioxin of Allinol exposed birds, the birds with necrotic lesions were nearly double that of the challenged alone birds. In their particular challenge model, they don't have a lot of mortality, but they tend to have a lot of birds, roughly about 20 to 25% across most studies, having necrotic lesions. So in this particular study, they saw nearly a doubling of lesions. And as you're looking at severity of those lesions, just on numbers of birds on a scale of 0 to 6, you see somewhat of an uptick in the severity of those lesions of birds exposed to Dioxin of Allinol. I'm going to skip those real quick and come to this slide, because this is really their thought on how Dioxin of Allinol is having a specific role and function on necrotic lesions. We know that it's disrupting those tight junctions, those barrier integrity, and we know that in the case of the pig, it's very specific for the Claudin family of the tight junctions. We see there's a change then to actual nutrient uptake, an increase to plasma leakage, which then we think is then feeding some of that perfringens, allowing it to essentially proliferate within the gut itself and add nutrients then for proliferation. The other interesting thing is if he took out perfringens and just incubated it with and without Dioxin of Allinol, and you do not see a change in virulence of that perfringens, just in incubation alone. It takes that additional nutrient source to really kick it towards going virulent. So we think just because of this disruption to that barrier really is allowing then that increase to that readily available nutrient source that's then taking that perfringens a little bit more virulent. And I know that those of you beyond the second row cannot read this, so my apologies. I would point you towards this particular review that Gunther had written, looking specifically at roles of different Fusarium species, whether it be mast, DON itself, Hyaluronone, or other fumonisins, on role on effects to different infectious diseases in the case of the bird. Really nice review that he did. Essentially, much higher sensitivity, increased severity, and duration of pathogenesis that's occurring in different disease models, particularly as those affecting the gut. So back to one of those fundamental questions that I had, do they affect the pathogenesis of enteritis? At least looking at those realistic and sometimes occasional doses, we do see changes to those inflammatory markers. We do see an increased need to try and quench that inflammatory response, and the bird is not quite able to do so readily. We think that's partly why there's an extended duration of this pathogenesis that's occurring. That those tight junctions are very specific to the Claudin family. There are some presumptions that we're making, and part of those presumptions are how then it's affecting plasma leakage and blood leakage into the gut. We assume then that that's what's kicking in the proliferation to Clostridium perfringens. But in essence, does it really matter? And we are seeing at those realistic to occasional doses some increase in severity of cocci and necrotic enteritis. But there's still a lot we don't know. We don't have a lot of effects on and knowledge on how multiple mycotoxins are affecting these particular physiological and pathological responses, and how they play a role with other intestinal pathogens. We really don't know at this point. So that's my last tidbit of information that I had for you. And I thank you, Raj, and the BIOMIN crew for inviting me here this afternoon.